right, it's just about 1:03, so I'm going to go ahead and get started, and we'll let people trickle in as they come. First of all, I just want to thank you all for coming. We're really excited to be having this webinar, and let's get going. So my name is Savannah Tracy. I'm the Communications Manager at the Cleveland Water Alliance, and we're just really excited to be partnering with John Higley of ECO today for this webinar. I just want to let you know that the Q&A box is what we'll be using today. Your screen should look something like this. There's a Q&A button at the bottom, so please feel free to enter your questions throughout the presentation, and we will be leaving time at the end in order to answer them. So before we kick off, I'm just going to give a really quick overview of the Cleveland Water Alliance, just for those of you who might not be as familiar with us. I promise it will be very quick. So the Cleveland Water Alliance is a nonprofit organization. We collaborate with a cross-sector of partners to really try to accelerate implementation and commercialization of new innovative technologies in the water space. We're a water innovation cluster based in Northeast Ohio, but we work Lake Erie region-wide as well as in the Great Lakes. So some examples of our programming. First, we do a lot of startup support of startup companies and businesses through acceleration and incubation support. You may be familiar with our Erie Hat competition, which is sort of our flagship program. We've given over $200K to early stage technologies since 2017, and we'll be growing that into more competitions in the coming years as well, and different versions of that competition. We also work on accelerating Northeast Ohio and the Lake Erie region's water economy and that ecosystem. We have a really dense water sector here in Northeast Ohio, and we're putting a lot of energy around growing and maintaining that ecosystem so that it can continue to thrive. We're also working to establish Lake Erie as the first smart and connected Great Lake. We're working with a lot of different partners on that to get cutting edge tech out onto the lake and the surrounding watersheds in order to monitor and to understand our water systems here in Ohio. And we do a lot more than that. So there's more that I can talk about today, but we just don't have time. So I really encourage you to check out our website for more information about the kinds of things that we do. So with that, I'm gonna hand it over to John. John Higley is the founder and CEO of ECO, and we're really excited to be working with him on this. So John, if you wanna go ahead and start sharing your screen, I'll let you take it away.
Great, thank you, Savannah. I hope everybody can see me now. Okay. Okay, so thanks everybody for coming and listening to me jabber on about nerdy things. We have a few poll questions open, and if you want to start filling that out now, and then we'll close it in a little bit. So go ahead and fill this out and let us know where you are in the region. We have a few other questions too. Okay. Great, so it looks like we're talking to the right folks. Great Lakes is certainly where we are spending a lot of our work and efforts right now, so that's great. And I think we've got a couple more questions for you to fill out. Again, looks like I'm talking to the right people. Excellent. Thank you for filling that out. This really helps us out, by the way. Okay. Okay. Well, you've come to the right webinar. This is exactly what we're going to be talking about today, invasive species as well as natives, of course.
That's the last poll, John.
Okay, that's what I was wondering. I was just going to double check to see if that was the last poll. Okay, wonderful. Well, thanks, guys, and thanks for coming today. I'll go ahead and close out this poll stuff so I can just see the screen. Yeah, so I'm John. I founded this company to help protect aquatic ecosystems. I'm thankful to the Cleveland Water Alliance for having us. Savannah has been wonderful to work with, getting this set up and doing outreach and whatnot. Joining me also is Marie Ball, who's our business development director in the Great Lakes region and our business development director overall. She would like to say something real quick. I think you're muted at the moment, Marie.
Just wanted to say I'm so excited to be sharing a new solution for the health of our precious water ecosystem. And thanks for being here today.
Yep, awesome. Well, cool. So without further ado, let's get into the meat of it. And I'm going to start moving through this. So let's see if my slides will advance. There we go. So, one of the things that when we're dealing with aquatic invasive species is, once they become a massive problem, they're very difficult to actually get rid of. And this is why we often talk about prevention. It's kind of the most important piece of this puzzle is prevention. And to do prevention effectively, you have to actually catch the species early. There are early stage interventions that can happen. Typically for quagga and zebra mussels, and this holds true for many other populations as well, you have to have multiple introductions before they can actually start creating a self-sustaining population that continues to thrive and grow out of control on its own. And we see this all the time with lake preservation. You'll see spikes in environmental DNA or some early signs of life, but then the population goes away. And we can get into how that can be exploited for mitigation as we go through this process. Overall, there's massive return on investment to do preventative work and to avoid infestation of, quagga mussels or Asian carp or these kind of things that are really destructive to fisheries. There's this, I recommend this paper by Cunahan and Bolins in 2017. They come to this conclusion because they're interested in protecting the Columbia River basin, which is not really infested with zebra mussels at the moment. And the concern is how much more testing with the current technologies would they need to do to effectively protect it. And the figures they came up with are about 100 times larger than what they're doing currently, and they do a lot of testing in this area. And I think that really highlights kind of the value of new technologies and new techniques for effective prevention and mitigation strategies. So first, we'll talk about the treatment as well. First, let's talk about the diagnostic platforms and kind of what's available currently. And I'm going to be focusing primarily on dry synods, so quagga and zebra mussel, and then Asian carp, because that's what we're kind of what everybody's really most concerned about right now. So currently, physical capture is pretty common. You see that with divers going down looking for populations of mussels, things like that. Plankton tow net capture followed by microscopy. You also see electrofishing as a big thing for Asian carp and other fishing things. And as you guys are quite aware, I'm sure that these processes are quite labor intensive. And generally, if you're finding adults or veligers, or the larvae of the mussels, at that point, you're probably already have a full infestation, or it's likely that there's a pretty decent sized infestation. And that's simply just a matter of doing the math of how big of a water sample am I taking, and what are the chances of me reaching in and taking this fractional element of the water and finding the zebra mussel or Asian carp. It's a needle in a haystack type of situation. We want to find it when it is just a needle in a haystack and not all needles and a little bit of hay. My background is primarily in cancer research and biopharmaceutical development, cancer diagnostics. And, you know, this is the same thing that we see with cancer detection. We always say that, you know, prevention is cure. You know, early detection is cure. And the difficult thing when you're trying to find cancer early is you want to find something that's very rare in the body, you know, something that's not like all the other cells. Because if you find it when there's so much, you know, tumor slough in the system already, that's when it's already quite a large tumor. And this is the same thing with, you know, when people do palpations for mammograms and things like that. The issue is if you can already feel a mass, there may already be a problem. We want to catch it before that mass even arise. And that's what a lot of the science in that area is driving towards. And I think that you can utilize the same concepts and techniques for environmental management. So there's also, you guys are probably likely using environmental eDNA. This is typically a rather small water volume, and you can analyze it by traditional PCR, which I'll get into what that is, QPCR, or there's some isothermal PCR methods coming out now as well. We'll talk about what all those are. Currently, kind of the gaps in this is that they don't actually tell you if what you're sampling for is alive or dead. So if you get a positive eDNA hit for Asian carp, and then you go out and do a whole bunch of electrofishing, and you don't find an Asian carp, well, then that kind of was a wasted effort. You know, the electrofish, there's a gap there between what you're detecting and whether or not the thing is actually alive. I mean, because you can find DNA from, you know, something ate it and pooped it out. There's a slough cell. There's a dead thing. You know, the DNA, you know, flowed downstream. There's a lot of different ways to get signal without actually having a live specimen. There's a big gap in the detection area there. So, this is so weird trying to advance slides on this. There we are. So, let's kind of talk about what this stuff is. This is a really wonderful paper. Barnes and Turner kind of breaks down all the different elements of environmental DNA and in the ecology. This slide I know looks very technical, but I want you to focus over here in part B where it says state. Nucleic acids are water soluble, so, and they also bind or are attracted to, not a hard bond, but attracted to, kind of sticky, to certain types of elements and chemicals. And so the water chemistry is going to change significantly the longevity of the nucleic acid, whether it's DNA or RNA, and where it's going to be most concentrated. So when you're looking at free and dissolved stuff, this is just stuff that's in the water, floating, it's fully dissolved. Likely doesn't have a very long half-life. But these particulate matter ones and extramembranous and intramembranous, these are associated with some other particle or, you know, or a biological. So it could be a piece of sand because silicate likes to bind nucleic acid, or it could be, you know, an actual cell or something along those that nature. Now, when you take a water sample and you're sampling for this, and it's just a raw water sample, you really don't know which one of these you're getting, or you're probably getting a mix of all of them. And there's some impact on how that, on your sensitivity and specificity levels downstream with that. But ultimately, what we do at EQO mostly focuses on this intramembranous to particulate. However, all of this can be utilized. And I really recommend checking out this paper to understand the ecology of environmental DNA in a water system. So now let's talk kind of a little bit, take you back to Bio 301 and understand kind of what makes the difference between DNA and RNA. DNA is just deoxyribonucleic acid. The deoxy means that it's lacking that oxygen that's in the RNA, that OH group. And this may sound just technical and chemistry-based stuff, but this makes a huge difference. So the background, the part of the structure of DNA is a sugar. And on the second prime carbon with a hydrogen, it's much more stable, and you form these very pretty little double helices. With the oxygen, there's kinks and curves and things like that. That's reactive, and so you get these single helices. And the reason why, I know boring chemistry stuff, but the point being is that DNA is a highly stable molecule. It's very resilient. A close friend of mine works on ancient DNA applications, and they can stick around the environment for a very long time. RNA is generally unstable. It has a lot of secondary structure and breaks down. but there's a lot of variety in RNA. When I used to teach, I would, describe to my students this as like, DNA, RNA, and protein are the good. DNA, good, easy to work with. RNA, bad, really tough to work with, falls apart very easily. And proteins, which are just ugly because they have all kinds of weird structural things that we don't need to get into here. So in the environment, you have DNA is pretty persistent. RNA, there's some variability, but generally it breaks down relatively quickly. Dissolved RNA in the water column may only last 40 minutes or less. And to an extent, RNA is the messenger molecule between DNA and proteins, and it naturally breaks down quickly in the body as well. So this is why RNA has not traditionally been utilized very often in ecology, mostly because it's hard to stabilize it and stabilize it in the field. Now, on this note, just to make it clear what we're discussing here, we're only discussing eukaryotic RNA. So RNA in viruses is a totally different field. Retroviruses have an extremely strong capsid, and so it preserves the RNA much better, which is why right now you're seeing a lot of work in wastewater for tracking of COVID-19. But that's a different can of fish. That's a viral capsid RNA is much easier to capture and preserve than environmental RNA from invasive species and eukaryotic organisms. So PCR, what is it? Essentially, what you're doing here is you have big long stretch of DNA, or maybe it's chopped up a few times. The point is you've got DNA. You take these primers, which have nucleic acids, this little string of nucleic acids, maybe 12 to 20 nucleic acids long, that are complementary to the strand of DNA that you want to amplify. So you have one forward 5' primer that's going to go and make a copy, and one 3' primer that's going to make a copy that way. And this works off of the same polymerase system that you use for normal mitosis. Is just how you copy genes. But we're going to exploit that and make a whole bunch of copies of the thing that we're looking for that's a positive identifier for whatever it is, because this is a very specific sequence. So what you do is you basically do this in cycles. You heat up the samples so the DNA separates, then you lower the temperature and the primers will anneal to the parts of the DNA, and you have a lot more primers than you have DNA, so you don't get the DNA binding back together as much. Then you raise the temperature so the polymerase is active, makes a copy. Then you lower the raise the temperature again so it separates, and you keep doing this cycle. So what you get is a exponential reaction of amplification of this segment of DNA. So what this can tell you, so in traditional PCR, your endpoint, you're not measuring this as you go. So you end up with a specific band of a specific length that you can look at and go, yep, that thing was present because I was able to amplify it. This can tell you if the segment of DNA is present or absent in a sample. It does not tell you if the organism that shed that piece of DNA is present or absent in the system. It cannot tell you if the source for that was alive or dead. And it cannot tell you if that source came from, poop or skin or, or an actual organism. This is very useful in verifying morphological characteristics. So if I find an Asian carp, I want to make sure, yeah, that's an Asian carp. You can usually just look at it and tell, but maybe the little guys, not so much. PCR test is a great way to verify what it is. Same thing with zebra and quagga mussels. It looks like one, it feels like one. Let's make sure that it is one. PCR works great. This is also why this is kind of the standard test for COVID-19 right now, is because it's a gold standard verification. So QPCR, a quantitative and real-time PCR, a little different. Essentially, we're doing the exact same process, but in the middle, we still have our forward and reverse primer, but in the middle, we've added this probe. And this probe is also specific to a piece of DNA in the middle, but it's got these two fluorophores on it, what we call, they're fluorescent molecules. So we've got a reporter. This is one that lights up, green, blue, there's a whole bunch of different ones out there. And the quencher. And the quencher basically hides the signal of the reporter. So when they're in close proximity to each other, it lets off no light. When you make a copy, it releases the reporter from the quencher, and so now the reporter is free to fluoresce. What that allows us to do is each time a copy is made, we get a little more light coming off of that. And we can have a little instrument that basically is shining a laser through it and just seeing what the reporter dye is doing. This allows you to quantify, because essentially, again, this is an exponential reaction. So the more of the thing that you're trying to detect is present, the faster the amplification curve will go up. And if it's not present or very few of it, you're not going to see it until later. So this is a way that we can actually quantify how much is there. This gives us a lot more power, especially on the DNA front, because now if we're doing this over and over and over again, you may not know if it's alive or dead, but you are seeing trends because you're seeing quantification over temporal, over time. You can see, oh, wow, I sampled right after Memorial Day weekend in this lake, and sure enough, our quagga mussel DNA went up. I wonder if I have an infestation now that these irresponsible boaters have just been all over the lake. This is very useful for risk management. So for understanding when am I at risk and where is it most likely for this to come in? Because you'll see DNA, again, because these species have to be introduced multiple times before a full infestation can take place, you'll see spikes in DNA prior to full infestation. It's very helpful for risk management. Doesn't give you a full understanding of what's actually going on though. And as I mentioned, it's helpful for early detection, risk assessment, and also for mapping to understand where is the density of, say, Asian carp, where do we have the most of them or the most activity, or at least the most of their DNA. Usually there's some relation there. So one of the things that we're doing that's a little different is we're using that environmental RNA. We have a proprietary methodology of stopping the degradation of RNA at the point of collection. And so since RNA is produced by genes, DNA to RNA to protein, if we can catch that message as it occurs, we can determine, is it alive or dead? Because we know that it breaks down very quickly in the water column. And if we're seeing, a gene for metabolism that's turned on, then we know that it's metabolizing, therefore we know that it's alive. The same thing can be said for looking at stress induction. So if we're treating an infestation in a pipe of, say, zebra mussels, and we're using a chlorine or some other chemical that's an oxidizer, there's a series of genes, for example, the heat shock proteins, but they're, for all kinds of stuff. There's a member of that gene family that gets turned on when they're experiencing oxidative stress, so changes in pH. So we can start to see efficacy. And there's some indication that we may be able to start predicting spawning by doing this as well, because a lot of those egg production genes get turned on before you actually have egg production. So you can kind of do some interesting stuff with this to kind of really fill that gap between DNA and physical collection. And then additionally, there's next-gen sequencing methods that you can do to understand population dynamics better and how those populations are interactive. That is actually mostly a DNA methodology, but it's potentially possible to do it with RNA as well. And this kind of enables us for better testing and to really understand how to do it. We also have an automated collection system that does all the collection and preservation for you on site, and then the end user basically just pops out the sample cartridge, sends it over to our lab, and we just trade back and forth sample cartridges, so you don't have to go out and take the same, the kind of labor-intensive methodology of taking the sample, and going out on a boat or having to go out there every day. There's an automated system to do that we have open, but this isn't a sales call, so I'm not going to be talking too much about that. Let's keep on the science, but yes, we are. So this is kind of the autonomous remote capture system. This is what it looks like inside. Essentially, there are 12 of these sample cartridges. There's a couple of filters in there of different sizes, and we run about 2,000 liters through a sample cartridge. Once that's completed, this buffer that stops the degradation of nucleic acids is pumped in to preserve the sample. And then on whatever time frame is most convenient, they're removed out of the autonomous sampler and sent to our lab, and we're just trading back and forth cartridges. There's also obviously methodologies to do this in the field. This is Erica, our chief science advisor, taking a sample in the field with one of these things, and that's very useful for finding baselines. So for example, we have a client that wants to know what's going on in my lake, then we would go out and take a whole bunch of point samples around to kind of get a baseline to figure out what's going on, or in my river system, a whole bunch of base points, a bunch of baseline points, and then recommend, okay, well, maybe you need to monitor this spot consistently, like near a dam or a marina or an inlet or something like that. And then we can be strategic about where we place the automated samplers, where we want to get a lot of samples over time. So that's kind of how that system works. And then obviously everything comes back to the lab, and we operate under GLP, which is an FDA regulation, but it's just a methodology for practicing laboratory standards the best way possible. I recommend checking it out. Just look into good laboratory practice, GLP. There's a Title 21. Anyway, there's a regulation thing. It tells you how to archive samples, how you need to keep your records, laboratory notebooks, times, all where everything goes, and how you have to do record keeping and laboratory handling. It's just something that we've adopted. There's not really a standard in our industry for that. We've adopted this one because it works great for FDA, so why not for us? So essentially, I kind of want to talk about how we do this. We provide these reporting tools, and we're following this decision tree that's basically off of, so my friend Adam Sepulveda over at USGS, and we're on this interagency panel for environmental DNA. We're the only private company on that panel as an official member, and I operate as a science advisor there. So he was putting together this white paper that's referenced here. Again, please go check it out. And it's essentially like, they're trying to bridge that gap too between an eDNA signal and physical capture. So how do you bridge that gap? Well, they put together kind of a decision tree for monitoring. Based on that and some working with him, this is kind of the decision tree that we put together for how we manage a lake or river system. Essentially, we start off with doing, some pre-infestation. For example, you're going to start off doing eDNA sampling, consistent eDNA sampling. Over time, if you see increased DNA signal, then you're going to test for life and utilize eRNA. And the cool thing is that once you have the sample, you can reprobe it for whatever you want. The samples are, whenever we take a sample, we isolate the DNA and RNA from the same sample, and then we can go back and probe those as many times as we want. It's essentially like a genetic database that you can query as you have additional questions. So if you see no eDNA or low eDNA or no change, then you just keep doing the sampling routine. If you see increased DNA, go look for life, probe for eRNA. Negative eRNA indicator, go back to return to routine sampling. Positive life signals, then we're going to start going into more advanced eRNA signal sampling. If we see negative eRNA life signal, it goes away, and we can return to normal schedule. If we see life is present, spawning signals are negative, or it's just a very small amount, it doesn't look like there's any spawn, we're going to increase the sampling schedule, but here it's to monitor a little more closely, but there's not a major intervention at this point. If we see increased life, lots of what looks to be a lot of spawn happening, then we're going to do advise to do some kind of early stage intervention. Now, some of this we'll get into the treatments and what you can do early to actually have an impact, and it has been effective in other places where they caught infestations early. If for some reason that doesn't work, you continue to have increased life and spawning, then you're going to have to move into a more full open water eradication. This is something that we're working on, but it's not instituted yet. And then, kind of repeat the flow as necessary. So this is kind of a whole management strategy decision tree, and you're, more than welcome to it. I'm going to go through a couple of use cases where we've done environmental RNA. We're going to be talking about some Texas lakes and some work that we did in Colorado. And I'm not going to talk too much about this other stuff that we did, groundwater tracing and fish hatchery thing. And these are some of the new projects that we're working on as well. I am going to talk about this Asian carp tracing project that we're pitching right now. We're going to talk a little bit about, I won't get into some of these other ones, But this interspecies population dynamics one is real cool. This is something that we're looking at doing in Lake Michigan to understand how these different species are interacting together. And again, since it's a database that we can query for whatever, you can take the same sample and query for multiple different species to understand what are the relationships between these guys, you know? And over time, those questions should become clear as we gather more data. So this is one that we did in Colorado. Essentially, what's going on here is you have, this was a lake that was a negative control. We knew that there was no infestation here. It's monitored extremely closely. Every boat going in, every boat going out is deconned and checked, and there's a stickering system, and it's a very well managed lake, and they test it constantly. And they have these this is for quagga mussels, they have these decon stands at each point where the boats go in, and they're spraying off the boats as they go in, spraying them when they come out. There's a whole process for this. So what we found was pretty big spikes of environmental DNA that were specific to quagga mussels around in this lake, which looked like there was an infestation, but we knew that there wasn't. So we went back and we probed these same samples for environmental RNA, and there's no life. What was going on is that essentially their prevention methodologies were highly effective. They were spraying off and killing these, you know, basically they were just spraying quagga mussel genetic material into the lake, even though they weren't spraying any actual live quagga mussels into the lake. They were all, the hotspots were all near the where the decon stations were. That red one is near the marina. That orange one is another. I'm pointing at the camera. My screen's on here. Anyway, sorry about that. The orange one is another decon spot, and then basically the closest one we found was kind of near the dam, and it was precipitous drop off based on how close it was to the decon station. This is one that we did in Texas. This was Lake Somerville. We found the question here was they were getting consistent environmental DNA hits, but they weren't finding any larvae ever when they go out and do net sampling. So they kind of wanted to have a little more information. We found environmental DNA all over this lake, but we found a small, I mean, it's a small amount. This is something that looked very much like early stage. 40 picograms of target is not a lot. The signal was quite low, but it was enough to indicate that something was alive there. And we did also see some RNA over here by the big marina, but the detection was so low. It was a real signal, but it was below our standard curve. So it was an estimated value, and that was likely not anything that would survive. From what I understand, there was subsequent interaction by the agencies there in Texas, and this lake is not listed as infested currently. So that was an effective use case. One of the things we want to look at a little bit more is big head carp, Asian big head carp and silver carp, Asian carps. This is kind of a list of kind of like where we're most concerned about them. Obviously, them getting into Lake Michigan or Lake Erie would be hugely detrimental. The state DNRs and USGS have done a really good job of mapping out what rivers they're most concerned with. So we want to put automated sampling devices at a couple of these locations to basically increase the likelihood of finding these early without having to do these massive electrofishing campaigns as frequently. Essentially, this is the idea, installing in autonomous samplers at Calumet River and Sag Channel in Illinois, and a couple other ones. I'll make these slides available so you guys can go through this a little bit more, but this is another potential application. We can look for eDNA because they're doing eDNA for Asian carp sampling all the time anyway, but at least, now we can do it automated. That adds some value. Then the environmental RNA is a nice backup to give additional information about what's really going on there. And we can talk more about that down the road. So since I'm running a little over time because I got excited about science, I'm going to keep moving along. And again, these slides will be available. So currently, prevention is the best solution. The electric barriers are very effective. There's sonar, there's also sound-based barriers as well that are being somewhat effective. The electric barriers in outside of Chicago have been very effective, but they did find an Asian carp past the electric barrier. And there's been some concern about how that could happen. Awareness campaigns are great. The signage is very helpful. Education is always helpful. The early intervention stages, like here in Texas, we had Lake Waco, which got infested with zebra mussels, and very quickly they operated to get in tarping, to basically lower the dissolved oxygen content enough to where the zebra mussels were not able to thrive and take off, and that population was eliminated. Physical removal is obviously something that we do a ton of. There's divers and scrapers to scrape them off of things. There's fishing campaigns. There's the community engagement fishing campaigns are wonderful, where you just send out a whole bunch of people, these great citizen scientists and volunteers who will go out there and do a whole bunch of fishing to get Asian carp out of an area. Super helpful. The whoosh fish can always makes me laugh. Obviously it's designed for salmon. If you guys haven't seen this, look it up. That's actually the correct way to spell it too. And essentially it's made for salmon. The idea is that to help salmon go upward, we've had humans go in and interact and disrupt their normal migratory patterns, and basically just sends them upstream. But it can also potentially be used for Asian carp removal. Use some acoustics to drive the Asian carp in a certain direction that you want to go, and then just start shooting them through the whoosh cannon out into the outside of the water where they can rot and die. Check it out if you haven't seen it. It's pretty fun. And then there's also obviously the chemical and biological solutions, oxidizers, there's pesticides and those kind of things. Biodiversity exploitation is something that I'm really excited about. I think it's a really cool methodology of doing that. We'll talk about that one a bit. And there's also these targeted biotherapeutics and biopesticides that are specific for the species that they're trying to get rid of. We're producing one of those. We'll talk about that a little bit. And there's a couple already on the market. But they're not really for open water just yet. Anyway, so we're going to be playing in that space as well. So let's talk about biodiversity exploitation a little bit. So what is biodiversity? Biodiversity, there's a few different components, right? So we're talking about genetic diversity within the species. We have species diversity, how variable are the species, the different species. So this is intra-species within the same species. Interspecies, what are the different species in an environment? And the diversity of the ecosystem itself. Those all are technically part of biodiversity. Mostly we're going to be talking about intraspecies and how we can utilize intraspecies diversity to eliminate invasive species infestations. There was a great quote. Obviously, everybody's biologist has probably heard the Dobzhansky quote from the 1970s. Nothing in biology makes sense except in light of evolution, right? Lynch updated that quote a little bit, saying nothing in evolution makes sense except in light of population dynamics. So there's always this conversation about natural selection, and people almost use it interchangeably with evolution. However, the reality is that genetic drift plays just as much of a role, if not a larger role, in many species, especially ones that don't spawn super quickly, or they don't have a lot of gene flow in between them. And again, check out Lynch's work to understand how population dynamics affect population stability and ability to persist in the environment. So the issue here is that if you take from a mother population that's somewhat diverse, and then introduce it to a new ecosystem, there's a good probability that some of this, you just lost some diversity. And maybe this gene just doesn't survive, and now it's just stuck this way. We see this with quagga and zebra mussels, because if you have a boat that has zebra and quagga mussel infestation on it, and then you move to another area, it's likely that all of those are somewhat closely related to each other. So if you have an early stage infestation, and this was successful in Colorado, if you just manage the heck out of it and make sure that no new quagga or zebra mussels get into that lake, the population that was originally there will essentially inbreed itself to death, and then it just goes away. And this is a way to handle early stage infestations. There's also some companies right now that are trying to engineer kind of a GMO zebra mussels for introduction or GMO Asian carp for introduction that will breed with the population that's already there, but they carry a gene with them that essentially makes it to where the next generation can't breed. So this way you can break populations down by infesting their biodiversity with a poison pill gene, essentially. And that's another potential avenue for remediation to go down. And the other cool thing is when you're looking at this, you can, if you're analyzing this on their genetics, you can understand how these things are spreading. I have to hurry up here. So another way is to, obviously, the chemical and biological treatments. Chlorine and other oxidizers, traditional coppers, potash. EarthTech QZ falls into this. It's much better than traditional copper. This is a, this product, EarthTech QZ, is a copper that can be dosed very low. It's an activated form of copper, so it doesn't have as much off-target effect as traditional copper salts. There's semi-targeted biobullets out there. Zequinox is a product that's been on the market for a bit now. It's essentially a bacteria. It's a subspecies of soil bacteria that produces some protein that they're not quite sure what, that then impacts the zebra mussel with some level of specificity. And we're producing one as well, and this is essentially a biotherapeutic approach. It's a engineered protein that we deliver in a microalgae form, and it's engineered specifically with utilizing antibodies to only affect zebra and quagga mussels. It's the same work that I did, or very similar to the work I did at my previous biopharmaceutical company, where we were selectively killing cancer and nothing else in the body. This is the same idea, same concept, obviously tweaked for environmental applications. So with that, I'm going to open up to Q&A. Just finished under the wire. Wanted to give you guys a full 20 minutes, but we got 15, so that should be close enough. This is how you can contact us. Please feel free to go to our website, submit through there. You can email Marite or myself as well. I will say Marite is much better about getting back in contact with people than I am. She's very consistent, which is one of the many reasons why we hired her. Or you can just submit through the website, and it'll go to our email as well. Thank you so much again, and now I will take questions. Thanks, everybody. So we are not having to be able to hear anybody.
Great, thank you so much, John. That was great. So we've got a couple questions here. I encourage you to continue to answer or submit questions through the Q&A box or the chat as well. I'm looking at that as well. So the first question for you is from Angel, and that's: are samples collected throughout the water column? Can the autonomous sampler be programmed for collection at different depths and times?
Yes. So the current version of the automated sampler is it's fixed depth. So what we do is basically determine what's the depth that is most likely to have the species that you're interested in or that you're most interested in, and we do it around that area. Obviously, it's pulling upward, so we're getting some suction from below it, but it is fixed depth currently. However, there's no reason why it can't be variable depth sampling. It's just an engineering issue. So if that's a necessity for a project, we would talk to our manufacturers who are kind of the tinkerers, and they like to do those kind of projects anyway. And even if it's a one-off thing, it's something that can be done. We're a small company, so these things are made on a as-needed basis. So if that's a necessity, then that's what would happen. And something that we've discussed as well. Let's see. Yeah, I hope that answers the question. As far as times, yeah, that's just programmed, and we program these things over a cellular network. So you can program the time of sampling quite easily.
Okay, great, thank you. Another question from the chat from Grace: are there any special techniques for collecting / field processing water samples when sampling for RNA due to the quick degradation?
It's not so much about the collection, it's about the preservation. So there are some handling issues just because, well, we'll go through kind of real quick how nucleic acids break down. You have kind of UV breakage, so you want to collect from, you don't want to have it exposed to the sun for a super long period of time. Temperature breaks down just because the enzymes at higher temperatures are more active. Enzymatic, so you have other proteins that will eat up break RNA. Metals can cause kinking in it that makes it fall apart. There's a whole host of these things that can make RNA fall apart. It's super silly and fragile. So what we do it with the filter sets, because this allows us to concentrate it. Since we know that it's breaking down relatively quickly, we don't want to take just a 1 liter sample. We want to take 2,000 liters to make sure that we can increase the sensitivity, and that increases our probability of capturing what we're looking for. And those are kind of that's kind of the rub on how to do that. Now, as far as doing nucleic acid sampling, there's lots of different ways to do it. And quite frankly, as long as you can get the nucleic acid collected in a way that is sterile and nuclease free, and then preserve it relatively quickly by adding the appropriate preservative buffer, you can do this effectively. The issue with doing whole water preservation is that the buffer has to be the main thing in there, right? So if you have like a 1 liter of water and then I'm adding in some buffer to stop the degradation of RNA, well, then I'm just diluting out my buffer and it's not going to be effective. It has to be at a certain concentration, which is the reason why we pack it onto those filters. Hopefully that's helpful.
Great, thank you. Just a reminder, please keep submitting your questions. We've definitely got enough time here for some more. So another question is, can the RNA detection of live or dead also quantify the number of organisms present?
So to an extent, the issue is there's a lot of variables there. And so connecting the amount of RNA or DNA that we're recovering to the exact number of organisms, there's so many variables in there that you have essentially it's too much variance. There's too much error in that to really understand, okay, if I have this much, if I have X number of nanograms of RNA that I've captured, that means I have X number of specimens, Asian carp or whatever in the system that I extracted them from. It could be one giant Asian carp, like a big guy, a whole bunch of little ones, and the variance in how I'm collecting it and the time that I'm collecting it, where they were in relation to it, all of those compound to create an error that you can't really make that one-to-one connection, unfortunately. Or even I would argue, I don't think you could make a really a range that would be meaningful. But you can see changes in content. But looking at this over time, you can certainly get an understanding of kind of relative levels. Absolute values, not so much. Relative values, absolutely. And so I think that relative value comparison is what's needed for appropriate mitigation anyway. So.
Perfect, thanks. Another question was, are there any plans for aquatic flora as well? For example, Hydrilla verticillata.
Yes, that is certainly on our menu. Erica, my co-founder, whose image was in the presentation earlier, her PhD is in plant biology and molecular ecology. So, and algae is something, algae or hydrilla, all those things are something that she absolutely wants to get into. The difference would really only be in the where you place the pump. Making new probes and making new probes for specific species or specific genes is not very difficult for us. We've got a pretty cool dual algorithm that we utilize for that, and we're querying databases pretty efficiently. It costs us about $8,000 to create a new probe for a new species or a new gene, which is pretty cheap. So yeah, it's certainly in the works, and there's not really, we have to validate it for plant material because we haven't done that yet, but it's certainly something that can be done. We've done it with cyanobacteria effectively. We have not moved into eukaryotic plants though, and that's something that we certainly would love to do.
Great. And this is the last question. So if you have a question, please submit it. We do have time for more. This is the last one I've got on the docket, but we can answer more. So Angel is asking, are there specific QA/QC procedures for the sampling for DNA and RNA? Are there blank or trip samples taken, for example?
Yeah, so yes, so sorry, I got distracted by another question that just popped in. I'll close that and rely on Savannah instead. QA/QC procedures, yes. So on the collection side of things, we validated that process. We don't take blanks, but we have a lot of, most of our quality control steps are in the lab. So we have control samples that we've banked from areas that we know what the makeup of the lake is, and we compare samples to those. So we have what we call kind of our limit of detection. So a lake that we know has no active zebra mussel infestation, but we'll utilize that. So, and gosh, I wish I had I could pull up my other presentation going through all the different controls that we have in there, but we do no template controls. We do known negative controls. We do limited access controls. We do internal controls for kind of like going after like a background gene that we basically spike in a little bit of DNA that we know what it is, and we amplify that and make sure that that's consistent across the board. And there's a few other controls that we add into this as well, mostly positive controls, negative controls, and standard curves. On the collection side of things, we're just collecting the water and preserving it there. And we validated to make sure that that does that over a variety of temperatures effectively. So I hope that answers it. Most of our control procedures are in lab.
Perfect. From Michelle, we've got you listed a few funding agencies for your work, including NSF. Have you considered applying to DOE funding calls, specifically the SBIR grants?
Yeah, we've looked into it. It's interesting. Actually, the building I'm in right now is Capital Factory in Austin, Texas. And the floor I'm on right now hosts the Army has their advanced development, kind of their new, I can't remember what the name of the core is, but it's kind of their innovations group is here. And I've asked them multiple times, like, what's available for this kind of work? And essentially it always goes back to talk to USACE or ERDIC. I'm just not as familiar with the Department of, oh, I'm sorry, you guys are talking about Department of Energy. I'm over here talking about DOD. Anyway, so DOE, Department of Energy. Honestly, we haven't looked into it that fully. We mostly focus on NSF. I'm more familiar with NIH and NSF, so that's where I went to for funding. And the Bureau of Rec, was kind of a happy coincidence of meeting the right people. So we haven't, but we should.
Great, thanks.
Sorry about that. I totally got confused and started talking DOD stuff.
All good, you answered it. So,
Yeah.
Okay. Is the RNA detection species specific? Can it be tailored to detect multiple target species at the same time?
Multiple target species, we would do different probes. It's specific for the gene and for the species that we're looking at the gene in. This is kind of the difficulty of designing these probes, is that you have to make sure that you're specific to the gene and the species, right? And so there's great databases to query to design these for intra-species within the species, and there's great databases to query for interspecies, like what is the species. We have this dual system that kind of scores those and brings those together. But we prefer to have everything specific. Now, we have designed probes that are just specific to dry synod, right? So, we have one that does zebra and quagga mussels both. We have one that does just zebra mussels, and we have one that does just quagga mussels. And that was just part of validation and kind of fun. But you can kind of design around that. If there's a specific application where you need overlap, it's a possibility. You'd have to look at the actual gene to see if it's possible to design it in a way that's restricted, but also a little open. And those are just design issues that sometimes are possible, sometimes they are not.
Great. One from the chat is, are you looking for test sites for the aquatic flora, example hydrilla, DNA/RNA testing? Does it get complicated when it comes to hybrid species for DNA/RNA testing?
It does get complicated with hybrid species because, yeah, hybrid species can be difficult because then you're increasing the issue of how do I get interspecies specificity and gene specificity. Like if I'm just looking for species specificity, even with a hybrid, or like there's certainly some hybrid hydrillas, even if I'm looking at a hybrid or a individual, there are ways to do that where you can be species specific. But if I then also only want to look at the gene of interest, sometimes that hybrid species and those two species, there's just no daylight in between that specific gene, especially when it comes to something like that doesn't evolve quickly. Like it can be very difficult, especially on the RNA side. Now on the DNA side, it can always get funky with introns. You know, introns are the segments of DNA that don't code. Those change. You know, you can get a little creative and do DNA work with introns to be very species specific. But on the gene specificity, it does get difficult with hybrids. Often possible, but it would need to be looked into a little bit further. We have not looked for test sites for hydrilla. Currently, we're focusing most of our efforts in the Great Lakes for zebra, quagga mussels, and Asian carp because that's what we are validated in, and we're moving into additional species as needed. Like if there's a project that will fund that work, we're happy to do it. But also, we're a very small company, so we have to maintain our focus. And retake and regale you with our sales and marketing strategy all day long if you would like.
That leads us into our next question. Actually, I think we've got time for it quickly. If you're comfortable telling everyone, what is your company's revenue model? Product sales release, service for analysis?
Yeah, so we operate again, we're in Austin and we're a startup, so we like to say we're a SaaS company because that's something people understand here. But we mean science as a service. It's essentially like a CRO. So we ultimately consult on the project. What are the needs? What's the method that's going to be needed? Work with the client to figure out how do we answer the questions that you need. Often there will be a baseline work where we go up and do point sampling all around, and that's either contracted to where we'll go up there and do it, or we send you some kits. And so you just do those individual samples and send them over to our lab. And then once we do the automated sampler, it's sold as a subscription. So you're not purchasing the equipment. There's no CapEx expenditure. It's a service agreement. You know, there'll be some upfront cost to have it installed and to have the training happen. But after that, it's just a, X number of dollars per year. You get 100 samples per year. You know, this is the time frame and go from there. And we have kind of a range of what that cost is depending on what genes are needed and frequency and how many samples and that kind of stuff. But then it's just, it's a Netflix subscription at that point. And as far as like our sales strategy, essentially we're talking to a lot of channel partners. So people like, we have an MOU with SWCA, which is a really great environmental consultancy firm, groups like that. Because, since we're small, getting into things like Lower Colorado River Authority, it's a big agency, and we'd much rather go in with a friend that they already know and subcontract through them than go in as, these kids off the street with this cool new technology.
Perfect, thank you. All right, I think that's all we've got time for today. Marate just put her email in the chat, so if you would like to follow up, feel free to do that. We'll also be sending out the recording as well as further information on how to get in touch. So with that, I think we're good to go. So thank you so much, John. It was great having you, and this was a really great presentation.
Wonderful. Thank you so much, Savannah.
Thank you. You guys have a great day.
Everybody for being here.
Yep.
Bye now.
Bye.